Welcome back to MCD 170, Society and the Brain. This is Lecture 8, which corresponds to Part 2 of Module 4. The title of Module 4 is Depression is Depressing. In Part 1 of Module 4, we focused mainly on neurobiological aspects of depression. Here in Part 2, we'll continue with neurobiological aspects and move into psychological and social aspects of depression. We ended off Part 1 by discussing the relationship between stress and depression and its possible mediation through the serotonergic nervous system. We noted that stress activates the HPA axis, which increases cortisol secretion, which decreases expression of 5-HT1A receptors throughout the brain. So we established a link between stress and cortisol and the serotonergic nervous system. But do these changes lower serotonergic tone? Probably not. Why? Because 5-HT1A receptors are inhibitory, and cortisol and stress reduce them. So reducing 5-HT1A heteroreceptors reduces inhibition by serotonin, and reducing 5-HT1A autoreceptors increases serotonin secretion. So stress through cortisol and downregulation, decrease in the expression of 5-HT1A inhibitory receptors probably elevates serotonergic tone. So the link between stress, cortisol, and serotonin probably cannot account for depression. Well, what else does? What could be the neurobiological basis of depression? Well, it turns out that stress and corticosteroids like cortisol cause reduced hippocampal volume due to loss of dendritic arborizations and reduction of neurogenesis. Neurogenesis is literally the birth of new neurons in the hippocampus. Genesis of new neurons, happens at a low rate in the hippocampus, but it's continuous. So over time, neurogenesis contributes many new neurons to the hippocampus. Depression is associated with reduction in neurogenesis. It's also associated with reduction in the processes, the dendrites and axons of existing neurons. How does this happen? Well, both stress and corticosteroids reduce brain levels of neurotrophic factors, which including brain-derived neurotrophic factor, nerve growth factor, and neurotrophin-3. So depression causes these neurotrophic factors to decrease, but chronic use of antidepressants can reverse that. It can cause the production of neurotrophic factors to increase again. So what exactly are neurotrophic factors? Neurotrophic factors are proteins. Yet again, proteins. They're involved in promoting the growth and maintenance of neurons and neural processes. Two well-known neurotrophic factors are nerve growth factor and brain-derived neurotrophic factor. Here's a little experiment where the experimenters put some neurons, bunches of neurons, on blotter paper. And you can see on the top, there are these little nerve processes growing off this bunch of neurons. They treated this one with nerve growth factor and look at the abundance of neural processes coming off of this bunch of neurons. So the nerve growth factor greatly enhanced the production and the maintenance of neural processes. Nerve growth factor was the first neurotrophic factor to be discovered. Brain-derived neurotrophic factor, BDNF, was discovered later. BDNF is probably the main brain neurotrophic factor. The hippocampus, again, is part of the limbic system. Here's the hippocampus shown here. And its associated dentate gyrus and parahippocampal gyrus. We'll talk about these later on in the course. The hippocampus is associated often with memory, but the hippocampus and related regions, dentate gyrus and parahippocampal gyrus, are important for the processing of stress. If the hippocampus is impaired, processing of stress can be impaired, and that can lead to greater anxiety. So as we mentioned, in depression, BDNF goes down in the hippocampus. These are results from an interesting article by Krishnan and Nestler, which is a review article available to you for free download. Here's a fact of interest. Depression is associated with decreased BDNF in the hippocampus, but increased BDNF in the nucleus accumbens. What's the nucleus accumbens? The nucleus accumbens is part of the pleasure circuit. We haven't talked about the pleasure circuit yet. The pleasure circuit is a bunch of neurons of interacting brain regions that mediate the sense of reward and pleasure in humans and other animals. It originates from the ventral tegmental area. The ventral tegmental area is a brainstem area that produces and secretes dopamine. Recall, the raphe nucleus produces and secretes serotonin, the locus ceruleus produces and secretes noradrenaline, the ventral tegmental area produces and secretes dopamine. These are the big three monoaminergic neurotransmitters. The ventral tegmental area is going to project and secrete dopamine in many brain regions, limbic regions, prefrontal cortex. We've already talked about the prefrontal cortex. We talked about the hypothalamus. We've talked about the amygdala. We've also even talked about the septum. The nucleus accumbens is new to us. It's part of the limbic system. It's also part of the pleasure circuit. It is a key area in the pleasure circuit. Secretion from VTA of dopamine into nucleus accumbens is associated with pleasure, intense pleasure. Most drugs of abuse cause release from the VTA into the nucleus accumbens of dopamine. So the nucleus accumbens is associated with pleasure, with upbeat mood, with resiliency. So what we're finding in depression is that BDNF goes down in the hippocampus, but it goes up in the nucleus accumbens. Already this is complicating the theory that BDNF or decrease in BDNF is the cause of depression. To get more insight into this, experimentalists developed genetically engineered mice. These mice had a mutation in the gene for BDNF, and it produced a mutated BDNF protein. This BDNF protein, it was processed abnormally by cells. This is a cell here. Here's the nucleus. The BDNF was transcribed into RNA in the nucleus, translated into protein in the cytoplasm by all this cellular machinery. But because it's abnormal, it's not processed normally by cells. This mutated variant is not properly sorted and less abundantly secreted from cells. So cells with neurons with normal BDNF will secrete lots of BDNF. Cells with this mutated form of BDNF will secrete much less BDNF. What is the consequence for mice? Net66 knock-in mice with this genetic mutation, show more anxiety, but also more resiliency following social defeat. This seems incongruous. How can we understand this? Well, the mutation causes less BDNF all over the brain. Less BDNF in the hippocampus is associated with anxiety, improper processing of stress. But decreased BDNF in the nucleus accumbens is associated with resiliency. So because that must mean that increased BDNF in the nucleus accumbens makes us less happy and less resilient. It's a very, very complicated situation. Again, showing us how complicated is the neurobiology of depression. So if BDNF is not the answer, maybe we can follow the link between depression and regulation of food intake. This slide concerns three hormones: melanin concentrating hormone, orexin, and ghrelin. These are hormones active both in the body and the brain that regulate food intake, and specifically all of them are orexigenic, which means they're all pro-appetite. MHC, orexin, and ghrelin, all three of those want to make us eat. Interestingly, MCH in high concentrations is associated with depression. High levels of MCH are associated with depression. In contrast, high levels of orexin and ghrelin are associated with mood elevation, and they have an antidepressant effect. So yes, there's a link between depression and food intake control by the hormonal system, but among hormones that make us want to eat, some make us depressed, but others alleviate depression. Again, very complicated. I think this is interesting in the context of the fact that depressed people can present with different um, eating abnormalities. Some depressed people eat more, some depressed people eat less. When you understand how complicated is the relationship between depression and regulation of food intake, that heterogeneous kind of presentation among depressed individuals starts to make a little bit of sense, even though the whole picture is very, very complicated. What does this leave us with? Well, it leaves us with a lot of complexity and confusion in the neuroscience of depression. We discussed 3 points: the relationship between stress and the serotonergic nervous system, the contribution of neurotrophic factors like BDNF, and the relationship between depression and food intake control. And specifically, what did we find? We know that limbic and raphe neurons have receptors for the glucocorticoids, and they have inhibitory 5-HT1A receptors. And cortisol suppresses 5-HT1 expression in both limbic and raphe neurons. We know that stress and cortisol are associated with depression, but decreased 5-HT1A expression would let raphe neurons produce more 5-HT and let limbic neurons respond to 5-HT more strongly, and that should relieve depression. So, yes, there's a link between stress and cortisol and the serotonergic nervous system, but it's not one that makes us depressed. It's probably one that relieves depression a little bit and elevates our mood a little bit, presumably to help us deal with the stress. Concerning BDNF, cortisol decreases BDNF in the hippocampus, but increases BDNF in the nucleus accumbens, and both seem to contribute to depressive symptoms. Again, it's confusing. BDNF in the hippocampus goes down, and that impairs our ability to process stress. BDNF in the nucleus accumbens goes up, and ironically, that makes us less happy and less resilient. But depression is associated with movements of BDNF in different structures in different directions. Very confusing. Finally, concerning depression and food intake control, MHC, orexin, and ghrelin are all orexigenic hormones, but MHC is associated with depression, while orexin and ghrelin are associated with mood elevation. Again, opposite effects in seemingly similar orexigenic pro-appetite hormones. It's confusing every way you look at it. So what does this kind of data lead to? Well, it leads to the view that maybe depression can occur in different ways in different individuals. This is an opinion of Paul Gilbert in this article from Psychological Medicine. That's your reading from the week. That's the PDF download for the week. According to Gilbert, depression is best thought of as a toning down of positive affect. It's a heterogeneous disorder, but it seems to be a final common outcome for a wide range of stresses and personal vulnerabilities. Stresses and personal vulnerabilities. The personal vulnerabilities are largely genetic, genes. The stresses are largely environmental. So he's really saying 2 things in general here. He's saying that, yes, depression is complicated and it could result from abnormalities in many, many, many different neurobiological systems. But he's also saying that it has both genetic and environmental origins. So what's some of the evidence that depression has environmental origins? Well, let's look at some findings. There are individual differences in depression between inbred rodents. Most experiments on rodents are done in the same strain of rodents because they want the rodents to be comparable. They want them to be similar. So inbred rodents have very similar genes, but they can have very different susceptibilities to depression. Individual rats or mice of the same breed, of the same strain, can differ greatly with regard to their susceptibility to depression. What about humans? There's a high discordance between monozygotic twins. Monozygotic twins are identical twins. They have the same genes, but one identical twin can be prone to depression and the other not. There's got to be an environmental influence there. Finally, the chronic relapsing nature of depression. It comes and goes even in the same individual, same person, same environment, the same kind of situation can make that person depressed one time and not the next time. So the relationship between depression, susceptibility to depression, and genes is complicated. It's shaky. Yes, there's a genetic component, but it's only maybe half the picture. The environment is the other half. So how can we study depression in rodents? Well, we can't ask them how they feel, but you can tell if a rodent is depressed because it can be socially withdrawn and because it won't engage in activities that it once found pleasurable. For example, mice love sugary water, sugar water, like little babies. They love sugary water. And they'll drink it like crazy. They'll lap it up. But a depressed mouse or a depressed rat won't drink sugary water at the same rate they used to. It's still sweet, but it just doesn't give them pleasure. It's pretty sad. Even sadder are the techniques that experimentalists use to make rodents depressed. One is the forced swim test. They put a little mouse or a rat in a cylindrical pool without walls, and the animal swims around and it gets to the edge of the cylinder but can't get out, can't climb out because the walls are smooth. Or they put them in a 2-compartment cage with a door between the compartments, and then they electrify the floor in one part of the cage. The rodent is free to jump into the other part of the cage, but then they close the door and shock the animal again. Now the animal can't escape. Its only way to escape was through the door, but now the door is closed. After a while, it loses the desire to escape. It just sits there and gets shocked and expresses depressive features, behaviors. Different kinds of social interactions can lead to depression. This particular one is isolation, social isolation. This mouse wants to associate with another mouse of its same species, but it can't. It knows the mouse is out there, but a glass wall prevents it from interacting. This kind of social isolation can lead to depression. During COVID-19 lockdown, I think a lot of us can understand that. Here's another mean one, social defeat. Now you put a little mouse in a cage with a rat or a mouse from a species of bigger mice. And this poor little guy experiences social defeat, kind of gets brutalized or beat up by the larger animal. Social defeat is a surefire way to make an animal depressed, including humans. So these kinds of experiments on rodents and on humans lead to psychological theories for depression. And again, these are from Paul Gilbert, you're reading for the week. These are psychological theories. The first two are non-social, the second two are social. And what Gilbert is trying to argue here, and you'll find more of this information in the reading, is that depression evolved because it might have adaptive value. Just like physical pain, if I'm in physical pain because you poke me in the hand with a pin or I touch a hot surface, I'm going to withdraw physically to prevent injury. He's saying that depression, anxiety and depression is like a psychic pain. And in our withdrawal response, we protect ourselves. So the first theory is learned helplessness. It says stressors, which at first elicit invigorated responses, such as anxiety and anger and trying to fight your way out of a bad situation, if not removed, can lead to withdrawal reactions. Incentive disengagement, a reduction in positive affect, in upbeat mood and energy, that enables animals to disengage in behaviors when the probability of achieving a desired outcome seems low. In both of these cases, something depressing happened. We can't escape the shock. There seems to be nothing we can do to improve our situation. The thing that we're trying to accomplish seems impossible. So we become depressed and we disengage and we withdraw. In both cases, we're not achieving our goals, but we are saving resources and we're saving energy. I guess it's the best of two bad outcomes. In the more social, the first one is attachment. Toning down of positive affect occurs on separation from the mother or significant others, so the cries for re-engagement will not expose offspring to predators. Well, if you're a little baby animal and you're alone, separated from your mother or from the rest of the troop, you're going to cry out, but you only want to do that for so long. If you're crying out and your mother doesn't hear you and the rest of your troop doesn't hear you, well, maybe the hungry wolf does. So you're going to quiet down and protect yourself. You might end up alone, but at least you won't be attacked by the wolf. Again, it's sort of the least of two bad outcomes. Finally, and most important, is the social rank theory, which says that depression is posited to result from involuntary social subordination or defeat. What is the evidence for the social rank theory? Well, depressed-like states occur in humans and other animals who have been socially defeated and lose social rank. It's probably easy for all of us to understand. Maybe once you were popular and had lots of friends, and suddenly somebody comes along who puts you down and changes the way everyone else in the group feels about you. And suddenly nobody likes you anymore. That's pretty depressing, pretty much depressing for anybody. That happens in humans and in other animals. And it can be studied in animals, and we can look at the neurobiological consequences. For example, subordinate baboons are more stressed, have higher blood cortisol, and take longer to recover from stressors than dominant baboons. So when you get put down, your cortisol goes up and you're not as resilient anymore. The dominance displays of dominant monkeys can lower the serotonin concentrations of subordinates. A big monkey puts down a smaller monkey, and that smaller monkey's serotonin level goes down and makes them less content. Social subordination in monkeys causes downregulation of dopamine D2 receptors and dysregulation of the HPA axis. Yep, that's similar to this one. And that cortisol goes up, dysregulation of the HPA axis makes cortisol go up. But downregulation of D2 receptors, dopamine receptors, makes you less responsive to dopamine. Remember, key pleasure interaction is ventral tegmental area secreting dopamine into nucleus accumbens, mostly onto D2 dopamine receptors. If you take away the dopamine receptors, your dopamine doesn't give you pleasure anymore. It can lead directly to anhedonia. Here, social subordination in monkeys causes downregulation of dopamine D2 receptors. That is one neurobiological basis of anhedonia. You can get socially put down to the point where you just don't enjoy life anymore. It's pretty rough. In humans, the subjective experience of social defeat was a more powerful predictor of depression than hopelessness. Among human beings, social defeat, social subordination is one of the most reliable predictors of a depressive outcome. And this can have important socioeconomic consequences. Before we get to that, what could possibly be the adaptive value of depression due to social defeat and social subordination? Well, depression in response to social defeat might have adaptive consequences in that the downregulation of positive affect and discontinuation of pursuit of goals signals to dominance that the subordinate is not a threat. Isn't that nice? We get put down by the dominant members of our group, and that causes us to be depressed and to withdraw from the group, to signal to the dominance that we're not a threat anymore. Maybe that has adaptive value. I guess if the dominants were going to beat you up if you didn't withdraw, then that had survival value. And indeed, some folks do respond to social defeat by becoming depressed and withdrawn. Others don't. Everybody's different. And the same person can be different under different circumstances and at different ages and for who knows what reasons. So yes, social defeat does have serious socioeconomic consequences too. because the subjective experience of defeat could be the result of direct interpersonal conflict, like, when you're in grade school or junior high or high school and your group of friends starts excluding you. That'd be kind of direct interpersonal conflict. Or indirectly, as failure to successfully compete for social resources such as caring, supportive or sexual relationships, meaningful employment, and material resources to avoid the stresses associated with poverty. These are some hard facts, and then they explain why lower socioeconomic status is linked to depression and suicide in humans, especially males. So what can we do about it? Well, we can take SSRIs, selective serotonin reuptake inhibitors, which over time, if you take them for long enough, a month or 6 weeks, will raise the serotonin level, and that can alleviate the symptoms of depression, but only in some people. SSRIs are only effective in about 30 or 40% of depressed people who take them. Serious depression is not effectively treated using SSRIs in all people. What about therapy? Various kinds of psychotherapy can help. The most common form of psychotherapy today is called cognitive behavioral therapy or some variation on cognitive behavioral therapy. It's based on the fact that our thoughts about situations can affect our behavior, but they can also affect our moods and feelings. More precisely, cognitive behavioral therapy, or CBT, is changing mood, the way a person feels, by changing thoughts about situations. That's how it works, in that direction. Its goal is to change mood by changing thoughts about situations. So with CBT, you explicitly change your thoughts about situations, and that can change your mood. If you respond to a situation in a different way, it might not make you stressed and anxious, and it might not lead you to become depressed. This is controlled and effortful at first. So cognitive behavioral therapy involves your controlled processes. Cognitive behavioral therapy involves changing your explicit thoughts about situations and using your controlled processes to control your automatic processes, using prefrontal cortex and anterior cingulate cortex to control fear and stress in the amygdala and hippocampus. And it's effortful at first, but it can become automatic and effortless with time and practice in some people. In some people. If you have a psychological impairment such that you cannot or will not change your thoughts about situations, then CBT is not going to help you. And cognitive behavioral therapy has been criticized and even lampooned on this basis. This is a cartoon illustrating that CBT can be ridiculous in certain cases. So drugs don't always work. Therapy doesn't always work. What's left? Well, depressed individuals who are resistant to drugs, resistant to therapy, resistant to the combination of drugs and therapy, which can work well together, will often resort to electroconvulsive shock therapy. These are clips from an old movie called One Flew Over the Cuckoo's Nest. This is the actor Jack Nicholson playing the role of Randall McMurphy in that movie. He was a troublesome guy, and they gave him a pretty strong dose of electroconvulsive shock therapy. Electroconvulsive shock therapy is still used today to treat difficult cases of depression. It can work. It also does cause a little bit of brain damage. It has to be repeated. The treatment doesn't last forever. So you might do a bout of a few treatments and be okay for a few years and then need a bout of a few more treatments later on. Repeated electroconvulsive shock treatments can cause a little bit of brain damage. But in seriously depressed people, it's better than staying depressed. What about kindness? No, you can't love somebody out of depression, but it's true that social support can ameliorate the depressive reaction to social defeat. And here's some facts from monkeys. Cortisol levels in subordinate monkeys are predicted by two key variables. A, the rate of conflict or stress. We've been talking about that in all of part 2. Stress, and especially social defeat, can lead to anxiety, which can lead to depression. Or B, subordinate opportunities for affiliative kin and supportive interactions. So just as social defeat can make you depressed, social acceptance can lift your mood, could alleviate depression. This finding is in line with studies showing that affiliative interactions, being friendly and kind, can stimulate oxytocin secretion, which can suppress cortisol secretion and can also enhance opioid secretion. Cortisol is a stress hormone. Chronic cortisol can lead to depression. We have endogenous opioids, the enkephalins and the endorphins. Endorphins get released when you exercise hard. They lift our mood. They make us feel good and relaxed, right? Oxytocin is a hormone associated with warm and fuzzy feelings. We're going to talk about it a lot in the next month. And the bottom line is kindness helps. It's not the solution, but it can improve matters. That doesn't mean that you can love somebody out of depression, as this slide shows, as this slide indicates. It doesn't mean you can love somebody out of depression, as expressed in this quote by Stephen Fry. But you can make the situation better for depressed people that you care about. Here's that artwork by Lynn Garga that expressed sadness. I kind of blocked the bottom in Module 1. In expressing sadness, she notes that life is complicated, but kindness is simple. I'll end there.